Welcome to this course on exploratory data analysis, ST9. So we're going to discuss different aspects of the introduction to this course in this first video on the topic. We're going to consider firstly the resources that may be useful in this area. There's a very good resource from the R for Data Science group on EDA, which is very useful. You know that that's in R, and we're using R in this course, but you may want to use Python. It's like a choice between tea or coffee. There's a good one on kaggle.com, which is linked here on an introduction to exploratory data analysis. And there's another one on deepnote.com. That's a beginner's guide to exploratory data analysis. There's two very useful guides on exploratory data analysis with R, where the Pang 2021 goes into great depth, going from the very start of learning R to very advanced modeling, whereas the Dylan Chills one is more concise and a more abbreviated version of the above. There's also a link to an article on this area which talks about dimension reduction methods and particularly with a view to omics data. So what we're trying to do in this course is to summarize and visualize large-scale data. And that means using dimension reduction techniques using multiple approaches. And we also consider how we scale, transform, and cluster data and how these methods all apply together. So we kind of want to really achieve these key goals, and they translate then to our learning outcomes. We want to be able to summarize and visualize high dimensional data, and we want to apply different dimension reduction techniques and approaches to understand why certain ones may be more appropriate than others for certain data sets. We also want to apply data scaling, data transformation, and data clustering approaches in the right way to tell us more about the data. So that's our goals. So when we move on, we can think about the course topics that are linked to exploratory data analysis. So in this first video, we're talking about an introduction to this area, principally focusing on this key question of why do we want to do exploratory data analysis anyway. That is then a sound basis then to think about an example data where we're looking at microbiome data, which is inherently heterogeneous, often analyzed in the wrong way and misinterpreted, and also is a great example of the intrinsic heterogeneity that many biological data sets have. That will then give us a basis then to consider dimension reduction, feature extraction, feature selection approaches to such biological data sets. And then we can think about how we can apply these certain methods in that area, such as principal components analysis, which is probably the primary one that's most widely used. Another complementary one is multidimensional scaling. In that vein, we'll also look at two other methods that are widely used, often used in biology, often misused as well. T-distributed stochastic neighborhood embedding, also known as TSNE, and the UMAP, uniform manifold approximation and projection. So what we're going to talk about next is where exploratory data analysis comes from. And that's John Tukey's 1997 book on EDA, where he's trying to consider how can we gain information from large data sets that help us formulate and refine hypotheses that will give us informative analysis or tell us about how we should, what samples do we need that will best inform our research hypothesis. So that can be linked to four key questions about that can arise from EDA or outputs that arise from EDA, such as having the right hypothesis that might be linked to certain phenomena of interest, to consider the selection of appropriate statistical tools and techniques, which depends, as per point 3 here, on the assumptions on which they can be based. And then also the fourth item is very important, that we may not have all the data that we need to answer these research questions, and therefore we want to collect more data. So that leads us on to thinking of EDA as simply a mix of numerical and visual methods, where the visualization is really, really critical to understand better and quickly summarize so we can decipher what's going on and is the data valid? That's the most central question here. Numerical methods also assisted in that goal by giving us a sense of what's interesting about the data as well. Typically, this is done before any formal modeling or any kind of particular test being applied, but that would depend on the data set as well, because sometimes there is some modeling involved, depending on what is our ultimate goal at the end. Please note that as well, you can't really do AI-related work unless you do EDA. First, because AI-related data sets are typically very, very large, very heterogeneous, often challenging to interpret, and we can't really get a sense of their validity unless we do EDA at the start. And of course, if we don't do proper quality control on data sets, we won't get decent results, and we won't be confident that they're actually correct. So often our output from EDA is that we find out that we need to do more data cleaning and we need to change our original sample set that we were looking at, and we need to repeat things again. And that's quite common and links back to the time that's spent just cleaning data, curating data, and managing data. That is quite normal in any particular project. And that's also links to using AI tools, that having AI ready data is really pivotal for that. You can't do AI related analysis unless you have those data sets. And that's why this course is the first part of a course on an introduction to applied AI. So the typical EDA questions that might be answered are: what are the most common values of a particular variable? So that could be using the mean, median, mode, different features in a quartile range. We might think about how much variation do we have in our data sets or groups. And we might also think about the tendency for different variables to be associated with each other or not. We must also think about what stage of analysis are we at and what are we trying to do. And often with EDA, it's very, very useful. It's very good because it's describing the data. So it's quantifying the basic features of the data that we have. So we're trying to understand numerically what we have. We're also trying to visualize it, because often there are quirks that we can only assess by visualizing this distribution. So that's also key, because we may find features that we don't expect, and that allows us to draw preliminary conclusions on our initial data that we can pursue more carefully next. And if you've taken these courses before, you'll know that these are available in the initial course and ST1, where we focus on simple ways to describe and summarize data visually and numerically. So another question that's related to this is what is your research question? What analysis or type of analysis are you trying to do? So with EDA, with any kind of high dimensional data set, we typically have really, really large amounts of data, and we can't or shouldn't test everything, because it's just not a rational use of time. And we should think about what are we trying to do and what stage are we at in terms of this hierarchy of different analysis. And EDA, what we're often trying to do is describe data sets as descriptive analysis, which is perfectly fine, or to summarize it so we can kind of decipher the key trends more easily. A more advanced part will be looking at trends or relationships that may generate a hypothesis, such as the correlation of plant growth rate versus the moisture level of those particular plants. So that's kind of, again, another key stage, and often a common one at which you might find yourselves when we're trying to analyze biological data. Inferential means testing if a pattern holds beyond the current data set. So it means that you've done your analysis of a given set of plants, and then you're exploring that hypothesis in a new set of plants. Could be plants that are published already, or ones that are being tested now. But that's often beyond what's incorporated in a typical project. Predictive means using the features data to predict outcomes. So, for example, if we have the association between plant growth rate and moisture, then maybe we can use moisture as a proxy to predict future plant growth rates. But we need very accurate and large-scale data to do this that considers all the variables that can affect plant growth. Then we have causal. This is identifying where they're changing one variable changes another, and proving moisture affects plant growth is an example of that. I guess what we're trying to track here more precisely is the rate of change of moisture affecting plant growth, and proving that this is not just an association but a direct causal link, which is quite complex when you consider the number of factors that can affect it. So we're really trying to think about all the different factors that can affect plant growth rate and measure the tendency of moisture to affect that and the other covariates. And finally, there's mechanistic. So this is looking at the exact underlying mechanism of relationship. So how does a molecule of water affect plant growth rate, and a molecular level, how does it get into the plant, get into cells, etc. All those steps are involved. So that's very, very detailed, and therefore it's useful for us all to think about what stage of analysis we're at. And this comes from work by Li and Peng, asking what is your research question, and what type of analysis that were you doing. So we can kind of look at this diagram here. The summarizing the data is always the first step, which is kind of what we're doing here as part of our course. And we're trying to essentially just describe the data. We may have for your project, you may have a new sample which is exploratory, but that is not often the case that we have that information. So we have to kind of think about this point from their work, and we should think about what we can say, not what we want to say. And it's very important. Often we want to jump down here to causality, but often we don't really have the data to say that. So that's the first major error, that we have a perceived question about causality, whereas we're looking at inference, and when we were looking at associations. But given the complexity of many phenomena that we all examine, we have to be very careful about that. Another is inferential, and looking at how one variable affects another, and assuming that there's a direct relation between that can be replicated in other studies. Sometimes this is referred to as data dredging or p-hacking. So that means applying lots and lots of statistical tests and then reporting the ones where the p-value appears to be small, which is bad practice and is, the perception maybe that is doing scientific work and doing inferential analysis, but in reality, it is not. It's just often doesn't consider the size of the study in terms of the power it has to make inferences. Predictive models are complicated, and often we need multiple data sets to do them effectively. Then this is to avoid the issue of overfitting. So when we link this to AI models, often we'll have testing and training data sets where we want sufficient samples in each so that we can separately check that our model is not overfitting or being designed in the wrong way, such that it loses predictive power in other data sets outside our own training one. Another common issue is that we're actually doing descriptive analysis when we just have one data set. We're not actually doing inferential analysis because we're not looking at multiple data sets. So these are all common errors, and there's many others as well. This is just a quick summary. So what can we do? We can focus on a specific research question, and often these analysis, the EDA will bring you back to this point. What is your question that you're trying to answer? Thinking about that very carefully and very precise and detailed terms will make things much more smooth and much more easy. And often that can be the hardest point, that people want to find out whatever is there, but that's not a useful starting point when you have a large data set. You need to be specific and focus on that particular question rather than trying to ask lots of questions. Another related issue is trying to mine data dredging, data mining, whatever you want to call it. And just looking at correlations between a bunch of different variables is not really is not useful. It's not an analysis unless you have some sort of unique data set where you can be sure that you've the full population that possess a phenomena, a particular phenomenon. Only then can you really be sure that you're looking at those particular associations are actually informative and reproducible. So when you see things like data mining and whatever, these vague terms, they're often linked to the point above where there's no specific underlying research question. So it's really not a good use of time. An example of this might be, are there more Bacteroides cells than Firmicutes cells in my cases? That's great if you have cases, but you need controls, etc., experimental design to test these things. So we think about more of a detailed question. What is the average normalized Bacteroides to Firmicutes ratio in these cases relative to my controls? That might be a more precise question where we can actually answer that in a scientific manner and interpret that appropriately, because there's a lot of steps along the way that we're looking at the BF ratio, we have to normalize it. Are we looking at the average ratio or the ratio for individual cases? And what about our controls? Where did they come from? How do we know that they're actually useful controls for this particular analysis? Another key question in this area is, do you have the data to answer this question? And sometimes that will be no. If you don't have an answer, don't have the data to answer a question, then we shouldn't try to answer it, and that will save time in the long run, even if you have to, uh, not deliver, you're unable to deliver what people want. An example of analysis and how we have to be careful about what we're doing and why EDA is important is that this example from the Public Health England in September 2020, when they were monitoring SARS-CoV-2 data, where they had an Excel error because they were converting CSV to XLS in Microsoft Excel, and there's a limit on the number of rows that are available, and for whatever reason they're arranging their samples across rows instead of columns, and they're trying to do this in Excel, which is utterly insane. So they had 15.8 thousand cases missed. And what they were able to plot then in this study, where they were able to look at the additional COVID infections in the 6 weeks period following the error. And they found that this is the kind of gives you a set of the percentage additional ones, but they're able to find that there was really a large number of additional COVID infections and also a large number of deaths also associated with that. So having, if they had done EDA on the process data they're trying to analyze, or even just did some simple quality control, they would have detected this issue, but they didn't. And this is the type of thing where just a simple quick checking of things, especially if you write a script and can automate these things, you can check that what you're doing is correct, and that kind of quality control is very, very important. So what can go wrong without EDA? There's an example from the Bagley and Coons versus the Potty Lab from 2006, and Potty et al. Have published work saying that gene expression signatures predict chemotherapy responses, and then these were used in clinical trials. So again, it has impacts on patients who are taking clinical trials for chemotherapy treatments. So this is very, very important and potentially can affect people's chances of survival versus death. Bagley and Coombs found that Potty et al.'s gene lists were off by 1 row, and that the training test tables were sometimes swaps, and in one case the sensitivity and resistance labels were completely reversed. These are really, really major serious issues, and the clinical trials were obviously were suspended. And this also links to other work on by Poor and Knight et al., where they looked at microbial analysis of blood and tissues that suggested cancer diagnostic approaches. And they had over 18,000 samples from the Cancer Genome Atlas across 33 cancer types on which they applied different machine learning models. They claimed to have identified microbial patterns in the tissue and blood differentiating different types of cancer. They even set up a biotech startup, Micronoma, making blood microbiome cancer diagnostic based on this work. However, they found Gehawi and Salzberg found major data analysis errors in this data set. Simply what they found was not correct because they didn't filter out human DNA. So many human sequences were classified as microbial, even though they weren't. And this meant that the cancer species were biologically not meaningful. An example of this was Hampadanovirus, a shrimp virus, which you can see in the image on the right-hand side, was apparently identified as being very, very important in adrenocortical carcinoma. So these kind of flawed, if you look at the details of these flawed pieces of work, you'll find that they're not really based on anything rational. But it's very important that we do EDA to ensure that we have taken the right steps. And microbial analysis and cancer analysis are very, very complicated areas that require very, very careful methods. So what was more serious about this paper is that they used normalized raw data against technical batch effects using VUM SNN normalization, which actually created a signature for each cancer type, but that was based on the sequencing center. Because they're organized by time, you had a batch of samples from a particular center, and because it was segregated by time, this cancer signature was derived essentially from the time/sequencing center. These are correlated, and that was the issue with AI-related models, that they tend to just take whatever data is there will be taken, even if it's not useful, informative, or shouldn't be there. And this led to multiple paper retractions, and the company halted. So this is why for our example data set, we're going to look at some microbiome data set, just for example, so we can see that it is complicated, and we need to think about it quite carefully. One of the things coming from the study, it was how we can think about different normalization methods. So, for example, when we look at different centers in sequencing outputs, so if we look at the original abundances and we do non-metric multidimensional scaling, NMDS, on that data, we'll see that there are clear artifacts where the level of mixing of the different groups is somewhat biased. You can see the orange on the right across the axis 1 of the plot, and blue are on the left, and purple are somewhat scattered across axis 2. There's different correction methods to adjust for these batch effects. And what we can see is that combat correction is good, that they're more close together, but percentile normalization is a more effective method in terms of the spread of these particular spots on this plot. Another example of this is when we look at the number of significant OTUs across different methods. If we do nothing at all and just take the non-normalized data, which is obviously biased because the relative abundances depend in the original study, we find that the number for that will be very, very high because many of these are false positives. If we look at the limit-based correction, it's still quite high. Combat is still high, but percentile normalization here in orange is more effective for doing that. And this considers the fraction of control samples from other studies. So trying to use the controls for different studies. So here we have Zeller versus Baxter, where we have, we're mixing, we've got Zeller, and Zeller versus Baxter here for different comparisons, and trying to mix together the controls to see how that affects our output. So that kind of brings us to the end of this video. Please follow the next video where we'll talk about statistical testing applied to the microbiome data.